Hey everyone, thanks so much for joining me on the BioBreakthroughs podcast, part of the Slice of Healthcare Network. I am joined today by the founder and CEO of Siren Biotechnology, Nicole Paulk. How are you?
Doing well, doing well.
And you have this sweet swag, as I pointed out, that looks like the softest sweatshirt ever. Am I right? Or is
you have to in San Francisco. It's so cold and foggy.
Well, there you go. There you go. I just realized I on this sleeve, so I'm going to have to wash this after, which is great. But hey, we're starting off on a strong note. Super excited to have you here. Let's dive right in to first, I want to go into, before you even talk about anything with the company, early inspiration, right? What initially drew you to the field of gene therapy in the first place?
So this all kind of started back during my PhD when I was at OHSU in Portland. And it was at a time when gene therapy was admittedly very unpopular, right? This was in the period where the FDA had halted clinical trials because of the death of Jesse Gelsinger, not from the particular type of virus that I use, but from another virus that was used in the field commonly at the time called adenovirus. But it was a period of time where literally you could not run a clinical trial, like federally, and you also couldn't receive federal grant dollars from the NIH to do this. But I was really interested in just the beautiful concept of it. Like, we're going to use a virus to replace a functional copy of a gene that you're missing, because of kind of the elegance of what it can do, right? Gene therapy to me is like the third wave of modern medicine. It enables something that wasn't possible prior to the discovery of that modality. So things like antibiotics and vaccines, right? They were able to do things that weren't possible prior to their discovery. And gene therapy was the same, right? You can't target something that isn't there. So if someone's born without a gene or has a mutation in a gene and doesn't produce, the functional product of that gene, usually a protein, right? If they're not producing that protein, you can't target it with a small molecule. You can't target it with an antibody. You can't target it with anything. Nothing can bind to something that doesn't exist. So the only way to treat these disorders was to actually give you a functional copy of that protein and of that gene back. And viruses are basically nature's little engineered FedEx truck. It's the best possible way to bring things inside your body and tell it where to go and deliver that package, in this case, a gene. And so I was really enamored with just kind of the ability to do something that could not be done by other modalities, even though the field wasn't very popular at the time. And kind of because of that, the great fortune of being willing to do something that wasn't popular was that all the kind of fundamental research in the field still needed to be done, because there were so few folks in the field doing the research at the time. And so there was a lot of just like really easy kind of textbook things that kind of needed to be sorted out about the field. And so the papers were fairly easy to come by. I did a pretty short PhD. It was about 4 years, but managed to publish 6 papers, which is a lot. Because there was just so much kind of fundamental things that needed to get sorted out and discovered. And so we were able to do that. And then the ball was rolling, right? Once you've published a lot in a field and you've gotten famous for it, then it's very easy to just keep going and do your postdoc in that and do your, become a faculty member of that. And so once the ball was rolling, I was hooked.
By the way, I don't usually smile on these much, but like your excitement is just getting me so even more excited for this episode than I was before. Not that I wasn't excited.
I've never been told I'm not loud or excited.
I love it. It's by the way, everyone watching this, if you want to know what loving what you do looks like, watch Nicole when she explains how she got into the field. We haven't even started talking about the company yet, so that's the exciting part here. Now, you and I were talking a little bit before about how obviously in the space that you're in, it's not uncommon to go from academia to entrepreneurship, but still, I would like to know maybe those early challenges, and then we'll go into Siren after, but what were some of the biggest challenges you faced in making that transition?
Yeah, I would say it's not uncommon for grad students and postdocs and maybe even undergrads, med fellows, not necessarily as common for a professor to resign. They usually keep their faculty position and then they sit on the scientific advisory board and be a founder of the company, but don't actually leave their role in academia to go start their company. So that is less common. But this happened at the peak of COVID. Remember back when you were still like Lysoling off your food that you got from Instacart, like those early days. That was when I decided to resign. And it was largely motivated by the fact that I wasn't allowed to come on campus into my own lab and do research, right? So we were at UCSF. This was a public institution, not a private institution. So they were beholden to state legislative laws and these types of things. And unlike Stanford University, right, which is about 20 minutes south of us, which was a private institution, which was allowed to, come up with their own rules, and they would let folks come back and do the 6 feet of separation and wear masks and gloves and these types of things. We weren't allowed to do that at UCSF. And so I was actually still kind of banned from campus for about the better part of 9 months. And this is, being a professor of virology. I study viruses professionally. This is only a BSL-2 virus. It's nothing. So it was super frustrating for me to know how to work with this virus and other types of viruses far more dangerous than this, and being told you can't come on campus and you can't work in the lab. And even when we were allowed to come back, it was 5% occupancy, 10% occupancy. And so it was just so frustrating to not be able to have folks come back, do work, right? We're a wet lab. So we need to be, remember the movies like Contagion and Outbreak, the people with their arms in the tissue culture hoods manipulating the cells, that's us. We can't do that remotely. You have to be on campus. So I was really frustrated by the fact that I couldn't go to work. And we had been sitting on the data that fast forward would end up becoming Siren. We had been sitting on this data for a number of years. It was at a point that was very, very exciting. And we were just sitting at home and reanalyzing old data and writing grants and, making a review article and all these things that are important, but that are not making new data and making new advances. And I was really frustrated. And realized that all of my colleagues that were out in industry were considered essential workers and they could go in. And I was just well, what if I just started a startup? And went and talked to the department, and they're well, theoretically you could start a startup, but you can't use your NIH, your federal grant dollars over at the company. You would have to go get VC money. That's separation of church and state. You can't be using federal dollars in a company. I was yeah, but assume I could do that. And they're but you'd also need space. You can't do any of that on campus. You'd have to be off campus in some kind of an incubator or something. And I was yeah, but assume I could get that. And they're you'd also need employees, you'd need permits, you'd need all of these things. We need to sort out the IP. And I'm yeah, but assume I could do all that. Could I start a company? And they're yes, I guess. If you could go do all of that, then yes, you could start a company and you could immediately go back to work and be considered an essential worker. And I was all right, I have got my marching orders. And I went and talked to, thankfully, because the gene therapy field at that time was still quite small. For the most part, there was about 5 of us who were professors who were famous enough that we got asked to do diligence for just about every single transaction that happened in the field, whether it was a financing, an M&A event, the hire of a CSO, an IPO, an in-license, an out-license, didn't matter. We always, all 5 of us kind of diligenced every single deck. So I knew quite a bit of VCs, enough to be dangerous. And so I just kind of circled back with some of my VC friends and said hey, what if I sent you something? And they quite literally were if you quit your job, we'll fund anything you do. And that became Siren. And so Founders Fund led the seed, and we quite literally, we moved really far. We moved across the street, literally, to an incubator that's really common for faculty spinouts from UCSF, only I actually left. And went over, quite literally, we put everything on carts and just wheeled them across the street, and and started the next week, and got to actually be back in the lab and running experiments again. But starting in the peak of COVID was a challenge, right? That was back when, right, the word of the day was supply chain, right? You'd place an order for some instrument that you need, and you might need to wait a year for it to arrive. So getting creative with how are we going to run experiments without stuff? Without instruments, without reagents. So that required some creativity, to say the least, but got it done.
The steps that you in talking with the university in order to say if I wanted to do this, that just shows that you're an entrepreneur right there, right?
Yeah.
Because it's when people keep putting out that blocker, and then, or it's always fun, and then you're just yeah, that's fine,
yeah,
I'm gonna hit that checklist. That's not a problem. Yeah, but they put it in front of you, probably in some cases, to are you sure? You have to do this, and then you have to do this. Yeah. So what?
I mean, there were definitely some late nights, right? Academia is it is the best possible cult. I love it dearly. I have many friends who are still in it, and I'm a big fan of academia. But we very much put it on a pedestal, right? This ivory tower. And you definitely drink the Kool-Aid, and you drank it all throughout undergrad and throughout grad school and throughout your postdoc and throughout your fellowships and all these things. So they're very good at making you feel like this is the penultimate job and there can be no greater job. So there were definitely some nights leading up to deciding to resign where it's like those like come to Jesus moments where you're like sitting in your bed looking up at the ceiling and going like, am I gonna do this? Am I gonna leave my job to go start this startup? So there was some hand wringing getting used to that idea, but ultimately made the jump and don't regret it for even 1 minute. Would never even consider for 2 seconds going back. Startups. I think the pace matches my personality and my style much more. So loving it.
I love it. Well, that brings us to Siren Biotechnology. So Siren is pioneering AAV immunogene therapy. Can you please explain what sets this approach apart from the traditional gene therapy approach?
Yeah, so kind of like I was saying earlier, the work that I did more in my grad student days back when I got kind of addicted to the field. So normally when we say the word viral gene therapy, we're saying I want to use a virus to deliver to you, the patient, a functional copy of a gene that you are either missing entirely or that you have a mutation in and you're not producing because you have that mutation. So that's classic gene therapy. And 9 times out of 10, we use a virus to deliver that functional copy of a gene because viruses are very, very good at getting inside of you. However, historically, we've only ever used this technique to go after rare genetic disorders where you're missing a copy of a gene or you have a mutation in that gene, and not necessarily more common disorders and not necessarily disorders that may or may not be genetic in their origin. And so we got very excited in my lab about how else can we kind of expand this platform of these viruses that are very good at getting inside of you? Like, what else can we use them for? And wanted to try to create something that would address one of the biggest challenges in gene therapy, which is the fact that these are very, very, very expensive. And when I say expensive, I mean the 5 most expensive drugs on the planet are all viral gene therapies. Many of these are north of $1 million. Many of them are north of 2 million, north of 3 million per dose, right? So these are very expensive. We've got gene therapies up near $4 million, right? So this definitely works scientifically, but this doesn't work when it comes to access issues. This doesn't work when it comes to scaling these things to try to go after a disorder that might have 2 million patients rather than 20 patients, right? Historically, we've also gone after very, very rare genetic disorders. And so in order for a company to recoup their costs to go after a disease with 20 sick kids, right, you might need to charge $3-4 million per dose to recoup all the costs that it costs to develop that. But what happens if you want to go after something like cancer or diabetes or heart failure or something where there's millions of patients, right? That won't scale, right? You can't, when the average American is covered by things like Medicare and Medi-Cal and Medicaid and these types of things, right? You would break healthcare. It's already broken, but you would break it even more if you were going to charge that much. So it just doesn't scale the way we produce these things and kind of the way we've developed them. And so in my lab, the like fundamental founding thesis of the project that fast forward became Siren was what we called the Universal Payloads Project, which was, could we come up with a payload that you could put inside of a virus that wouldn't need to change with every single disease you went after, right? Because right now, like if I treat disease A, hemophilia B, I can't use that same virus to treat someone who has spinal muscular atrophy because the gene that you need, the medicine that goes inside the virus, the gene, the DNA, right? Those are different across those 2 diseases. But if you could make a virus that had a payload that didn't change across 2, 3, 4, 500, 5,000 different diseases, now you could make that one drug and use it 10 ways to Sunday and it becomes more like Tylenol, right? You can just use it to treat all kinds of things. And so we started the Universal Payloads Project that fast forward became Siren. And kind of tried to ask the audacious question, like, are there payloads we could deliver that could do something of interest therapeutically across different diseases and tissues and developmental timelines and these types of things? And so got very excited by a variety of different kind of classes of payloads that you could deliver. So things like transcription factors and cytokines in particular are very interesting to us because they're very what's called pleiotropic, which is just a fancy word for does many things. They're involved in infectious disease responses. They're involved in oncology. They're involved in allergies. They do many, many things throughout the body. They work throughout your entire body. They work when you're a little kid. They work when you're an adult. They work in all your tissues. And so we got very excited about cytokines as kind of a class. And kind of wondered, well, what would happen if you put these inside of a virus? And then we made this huge list of all these ones we wanted to test, plus it's Silicon Valley. So obviously we wanted to do like an all by all comparison and make these huge libraries of combinatorial synthetic circuits. And then the list was too big. It was like, well, shoot, we can't make all of these. We have an academic budget. And then we realized rather than making just a single one of these and kind of going down the rabbit hole and testing whether or not it might work in different disease settings, we're like, well, let's just make libraries and then use them as pooled libraries and test them in different settings. And one of the settings that we included in that list was oncology, was cancer, because this kind of the way your body currently fights cancer, the tumor that you have inside your body right now, everyone listening to this, you have a tumor in your body, don't freak out. It's a single cell. You've had a tumor for the most part every day of your life. And the cytokines in your body are able to mediate a response that helps your immune system fight and find that cancer and chew it up while it's still a single cell. We've known this for the better part of a decade. This is like the basis of basal background cancer immunosurveillance. Many companies have tried to take cytokines themselves, the proteins, and deliver those to patients as a drug. The big challenge is that they have a really short half-life, like how long they're active for, right? When you got your COVID booster, remember that huge fever you got that was super intense? That was cytokines, but it probably only lasted about a day and then it went away. And that's because the half-life of these things is very short. It can literally be minutes to hours. So these things have a very short half-life on purpose, by design, right? We don't want you to have a fever for a month, right? You die. So we want these things to have a short half-life. But if you want to use these as a therapeutic, that's now your white whale. That's the thing you have to battle, is the fact that they don't act very long. But that's when they're in the protein form. But they're coded for in DNA. So if you can deliver the DNA form of these things inside a virus, then you'll just keep making more copies of it from within the virus. And then when the tumor cell dies, the virus dies. So you have this like built-in kill switch that controls the timing of expression. And so we got very excited about these as a class and developed kind of a new modality that we're calling immunogene therapies. So using the specific virus that we study, which is AAV. And using these to go after cancer for the first time. So this is the first ever universal AAV gene therapy. So an AAV that can be used to treat more than one disease. And it's also the first ever AAV gene therapy in oncology.
And where do you, by the way, your explanations, and I got to tell you, I think a lot of people will appreciate truly how excited you get when you're explaining. You're more likely to like truly hear someone when they are, and like, so having you as like a professor would have been like, I mean, we need more of professors like that.
And I get told that. They're like, oh no, you left. We need more professors like you.
Well, it helps your students just, keep the information that you're telling them. Yeah, they get excited. If they get excited, you're more likely to learn, right? I always used to, I mean, what are the like two biggest ways to learn something? There's like a positive reaction where you get excited, you're going to remember that memory, or there's a negative, right? That's going to stay with you as well. We prefer not to do that one. But
carrots, not sticks.
Yeah, exactly. So you mentioned, I want to go back to this a little bit because you mentioned, and I'm curious if this had any impact on where the company is at today. But you mentioned when you were kind of in limbo land in the beginning, right? During COVID, you were just reanalyzing data, like a lot of data. Does that actually, because that's not necessarily something your circumstance made you have to keep reanalyzing it. Was there any positive that came of that? Like that maybe, or would you say for the most part, like you were just spinning your tires during that period? Again, not your fault, but yeah.
Yeah. I'd say a little bit kind of in between. It was a little bit spinning the tires. I mean, anytime you reanalyze old data, you always find new things that you can plug into a manuscript or into a grant application or into a upcoming presentation that you're giving a PowerPoint or something. So it's not that it wasn't useful, but it definitely wasn't as useful as generating new data, right? Fail fast. That's true in both academia as well as industry. Like run the killer experiment. Don't run the experiment to like prove yourself right. try to run the experiment to prove yourself wrong, right? See if you can find a way to break the model, break the mold, break the idea that you have. And if it keeps working, then it's probably real, the insight or the discovery that you made. But try to run the killer experiment, try to break it. And that's kind of something you can only really do with new data, especially large data. And so I would say a lot of the stuff that we were doing during that like 9 months when we were banned from campus was like gearing up. We didn't know it at the time, but it was kind of gearing up to write grants for Siren. So, not, I mean, thankfully we got some VC dollars to get started, but we also started a very large grant writing campaign, which proved beneficial since very shortly after COVID, where there was this huge win, right? The market was crazy. You could just spit on a piece of paper and people would give you VC money. Right. Then the crash came. And so it was very nice to kind of have a lot of data and material already ready to put into grant applications so that we could go out and get some non-dilutive money to help supplement while the market was readjusting.
And what was like,
and it's still readjusting.
It hasn't stopped readjusting yet. When you kind of look at,
thankfully all of our investors are very happy with our non-dilutive grant money.
I mean,
it's a win
who wouldn't
for everyone.
be, right? That's actually what I wanted to ask you is, before starting this company, you obviously had experience with writing grants and getting that federal money.
Basically a professional grant writer.
Well, how did it change though? Like, so did you notice any major, obviously when now you're raising money from, I know you went to some people that you've known and you had that relationship already, but this is something that a lot of people coming from your world, right, are like, I am used to writing grants and getting those dollars that way. Now I have to talk to investors. What advice would you have to those people that are seeking to now raise that outside capital from, investors?
I think the advice I would give now would be different than the advice I gave when I spun out. Like when I spun out, that was when you could literally just like scribble a note on a piece of paper and someone would probably give you, somewhere between $1 and $10 million. They weren't going to give you 100 million, but they would give you enough seed money to get started in biotech, like kind of without data. You just had to have an interesting concept. It was so easy to get VC money that it would have been a waste of your time. And you probably would have been encouraged not to write grants by your VCs. They'd be like, if you just need more money, just come talk to me. I'll give you more money. Because people were just shelling out money so kind of effortlessly. Now it's a complete 180. Now I actively get sent grant applications by my VCs. They're like, hey, did you see this? Hey, did you see this? So if there were academicians who were thinking about spinning out right now, I would say, like, this is one of your superpowers. This is certainly one of mine. I'm a professional grant writer, essentially. So if I'm head to head against another CEO who's never written a grant before in your life, like, I'm going to eat you for breakfast. Like, I am going to write a grant that is so much better than you. Like, they'll be incomparable, regardless of how awesome your science is. It doesn't matter. Grantsmanship is a thing. It is a skill. And it is very separate from how good you are as a CEO and how good your data is and blah, 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 all these things. So I would say to other academicians, particularly more senior ones who tend to write grants, right? So things like postdocs and faculty and less so grad students and undergrads, like, that's your superpower. Harness it. Like, go out, see if you can get some VC dollars. That's a thing. But don't shy away from the non-dilutive grant money that's out there, because it can actually be more than what you would get from a VC right now. And talk to everyone, whether or not they have a grant, right? So there's the classic places like NIH for SBIRs and the Department of Defense and BARDA and DARPA, depending on the technology that you use and whether or not it might have a military purpose and these types of things. But also disease societies and foundations, different vendor companies. But quite frankly, I just ask anybody if they have a grant, even if they don't have a grant. I'm like, have you thought about starting a grant program? And you become this like monotonous little grant machine. And you just find this stuff, you seek them out, like seek and destroy. You find these grants in these weird random places, and you just apply for absolutely everything. And that's your little superpower, because no matter what's happening in the market, like, grants will be there. And so that's your secret stash of a way to get money, even if VCs think you're too early, think your idea is too risky, think it's too big and bold, think you don't have enough data yet, think you're too far from the clinic, whatever the reason is that they're not funding you as you initially come out of academia. if you can get grant money, then you can de-risk and continue to advance. And then when they do circle back to you later, the price is going to go up. So it'll be
I
their
love
loss.
it. And non-dilutive is the
And
yeah,
non-dilutive, which is the best money ever.
absolutely. As we wrap up here, Nicole, you've been an absolute delight. This has been awesome. I'm excited. I could talk to you for hours. Like this is just, it's at some point when I'm out in your area, I would like to do an in-person one, and like we could do like a fireside chat because we need more energy like this. I'm also going to point to your episode for future people that want to come on. I want this level of energy that Nicole brought today. Okay, we need more of this in the world.
Some of it's
Yes.
coffee. What's some of it's
Yeah,
natural?
I switched to what is it? Iced Americanos. It's there. They can
Light roast, people. There's more caffeine than that dark roast. Light
really.
roast.
I did not know
Yeah.
that.
The more you roast it, the more you burn off the caffeine. So light roast is your friend.
Oh, all right. Well, I need to do that then. You're right. Oh, okay. That's why I'm over drinking.
There's your
That's
nugget.
why it's
There's
not
your
doing
nugget
anything.
today. Light roast.
I feel like I knew that, but it's been yeah. All right. Well,
Light roast.
Nicole, as we wrap up here, I'm sure there's a lot, but if you had to pick one thing that really excites you about the company right now as we kind of head into the second half of the year, what is it?
So this is a big moment for Siren. So we're actually like, we're right smack in the middle of the transition from being a preclinical company to a clinical company. So we're right in the thick of like all the gnarly mess. We're making our, clinical grade purity virus. We're doing all of the FDA submissions. We're doing our IND enabling animal experiments. IND is the big packet that you submit to the FDA asking them for permission to do a clinical trial. So it's like, it's becoming real and it's meaty. And it's finally starting to be like, oh man, we're going to be like dosing a human being soon. Like this is legit. So I'm pumped for the switch from being a preclinical company to a clinical company here in the kind of like next 12 months. So we're going to grow up. We're going to have like big kid britches on. It's going to be awesome.
I love it. Well, at that point, we should have you back on. You can give us an update on where things are at because we'd love to hear it. Awesome. Well, thank you so much for joining me here today. Can't wait for future conversations and best of luck and never lose that energy. I love it.
I won't, I promise.